In this video we will discuss about some of the methods that are currently being used to study intra-tumor microbiota data. So one of the traditional method is by sequencing the 16S rRNA gene sequencing, and in this also the different versions of this different methodologies of the 16S rRNA sequencing has been developed, and some of them used one of the most commonly uh, known company in the sequencing is this Illumina, that can sequence this 16S RNA. And a brief representation is shown here, that a part of the 16S RNA is being sequenced along with the barcodes and the primers that can be used in the flow cells, or that are complement to the flow cells, so that this 16S RNA gene sequence can be uh, sequenced and analyzed. And the target for the 16S RNA is or the idea is to sequence the 16S RNA of bacteria. So the disadvantage of this technology is that they can sequence only the known, they can get the information about the known bacterial sequence, not some new bacteria that are not yet known. And in order to do the deeper sequencing, the next stage is about this metagenomic sequencing. It can sequence all the genes that are present within the tumors, so it will give us the informations about not just the bacterial sequence but the other sequence that are presented in the tumors, or the mutations that are presented in the tumors, you can say like this. And now focus has also been shifted towards to find out the locations of this bacteria. In order to do that, immunohistochemistry is been used. So here a antigen is used against a specific bacteria or the protein of interest. So since we are analyzing, our topic is about microbiota, so antigen is about the specific bacteria that can be used. Then secondary antibody is used to bind the primary antibody, and the secondary antibody is then conjugated with some fluorescence indicator. And this is, you can say, similar to the two-step Western blot, where primary antibodies bind to the antigen, secondary antibody is conjugated with fluorescence is bind to the primary antibody. It will give us the idea about the not only the qualitative idea about the protein or the bacteria that are present in the tumors, but it will also give us the quantitative idea. Next is about this fluorescence in situ hybridizations. In this case, the gene sequence that are complementary to the bacteria gene sequence is being used. So once they hybridized with their complementary sequence in the samples, it will give a fluorescence. The problem with this fluorescence FISH method is that there are a lot of non-specific bindings that occurs, and because of that very few probes has been developed in this fluorescence in situ hybridizations method. And this is a representation how it works. Once it binds to its complementary sequence in the samples, it will give us a fluorescence signals. So the detail is not shown here. You can see the details in a article where they discuss, or the main focus of discussion is about this FISH. Next is about this culturomics. In this case, the tissues or tumors that they get can be cultured separately in vitro, and then it can be analyzed in a controlled atmosphere, rather than directly use the tumors for analysis. And because in that case the amount of bacteria that are present within the detectable limits will get will not be recorded. But when we use the culturomics, we can scale up the amount, all the amount in equivalent portions, such that all of them or most of them becomes in the detectable limit of the instrument that we will use for analyzing microbiota. And the histology, I have forgot to mention what the histologies will look. Sections will look like this. Fluorescence microscopy section will look like this, and this culturomics. One of the examples that is shown here is to find out whether the tumors has gram positive or gram negative bacteria cells. Gram positive bacteria will have higher cell wall, higher thicker membrane, so it can stain to give pink color. It can be stained with the dye to give pink color, whereas this is the gram positivity. It can be stained with a dye to give the pink, or here it shows the purple color. But when in the gram negative bacteria, they will not be stained and it will show a different color or no colors. And thereafter, since with the advancement in this sequencing, we will get a clean output that can be analyzed using bioinformatics to find out the relative change in the different samples or in the tumors from different regions or tumors from different cancers. So that can give us a more insight about what are the bacteria that are present there and how they are related to it can be related to the other cancers. So this is some of the intra-tumor microbiota analysis methods that could be used or that are been used in the past also, and are currently also been used. If you need to understand more about these experimental methods or these methods, not just experimental, the computational methods also in detail, you can go through the reference that has been provided in the description. From there you will find out an appropriate cross-reference that described each of these methods in depth. That's all for this video. Thank you for your time.